Systems. I'm Scott Blackman. I'm in genetics and pediatric endocrinology at Johns Hopkins. I'm one of the moderators. My behind-the-scenes co-moderator, who's working very hard, is Dr. Susan Burkett, who's a pulmonologist at University of Alabama, Birmingham. I want to take a moment to orient you to the screen in case you're not familiar at this point in the meeting. This is a live session. On the right-hand side of the screen, you have a text box with separate tabs for chat and Q&A. We encourage you to chat socially throughout the presentation in the chat tab, but please submit any questions that you have through the Q&A tab. They are reviewed by Dr. Burkett, and after her review, I will read the questions to the speaker. We won't be monitoring the chat for questions to the speakers. We have 3 speakers today. Dr. Hua Ling at Johns Hopkins will introduce us to the Cystic Fibrosis Genome Project, which seeks to discover genetic modifiers of CF using whole genome sequencing. Dr. Marlou Bierlock at the University of Utrecht in the Netherlands will present the first results of the HIT-CF organoid study, studying the response to a potentiator corrector and amplifier combination, in which can be used to characterize ultra-rare CFTR genotypes. And finally, I will be our 3rd speaker. Today, I will present initial results from the CF Genome Project regarding cystic fibrosis-related diabetes. And with that, we can transition to our first speaker, Dr. Ling.
Hi, good afternoon, everyone. I'm Hua Ling from Johns Hopkins University. It's a great pleasure to be here and introduce you to an exciting project, the Cystic Fibrosis Genome Project, CFGP, which is a multi-phenotype whole genome sequencing resource. This study was a joint effort led by multiple groups as listed down here, and I'm presenting the work on behalf of the entire team. So the first question came to your mind, maybe, why do we conduct such a large-scale genetic study, given the CF causal variant and genes have already identified? So many of us know that people with CF exhibit large variation in disease severity, complication, and survival. Many studies have found that other than CFTR itself, genetic modifiers play a critical role. Therefore, the purpose of current study is to identify genetic modifiers of CF traits using the technology called deep whole genome sequencing, and at the same time, try to develop a protected resource for the research community. So the study was initiated in 2018 with samples recruited from 5 cohorts across 3 different groups located at Johns Hopkins University, University of North Carolina at Chapel Hill, and University of Washington. Each group contributed about 1/3 of the total samples. Approximately 5,200 samples were sent for sequencing at the Broad Institute, including intentional within and cross-site duplicates. The twin and sibling study at JHU was a family-based one, which enrolled individuals with at least 1 affected siblings, while the CF-related diabetic study was a cross-sectional study that enrolled individuals both with and without CF-related diabetics. The genetic modifier study at UNC enrolled people with extreme lung functions that are pancreatic insufficient, and the liver disease study enrolled individuals with severe liver disease. The F study from UW was a longitudinal cohort that enrolled individuals with early Pseudomonas infection. So the difference in study design and the recruitment strategies inevitably resulted in difference in some of the baseline characteristics. For example, here is a density plot of the birth year by study site. As you can see that samples from UW were younger and has less variation compared to the other 2 centers. And since we have the registry data, we got access to a large number of phenotype variables, most of which are quite comparable across those 3 sites. And here, the table gives you some of the examples. What I would like to point out here is, consistent with the younger birth cohort, samples from UW were also younger at the most recent pulmonary function test and has the highest percentage of alive by the year of 2017, while the sample from UNC have almost 100% pancreatic insufficient due to the way they enroll their study participants. And since pancreatic insufficient was determined by the CFTR genotypes, it makes sense to have a higher percentage of Delta 508 homozygotes. So as you can imagine, combining these different cohorts into a single project requires tremendous work in phenotype harmonization and data cleaning. And this applies to the genetic data as well. So here is a list of the files we received from the sequencing center. And before we can use it for real data analysis, we conducted a very comprehensive quality control. So for example, we started with the sample level QC, including quality and identity check. And then we move on to the variant level QC using both the VQSR and hard filters. The effectiveness of those variant filtration were further evaluated by concordance rate from all the study duplicates and monozygotic twins. And finally, we checked the genomic inflation of the QQ plot from the common variant GWAS analysis to ensure that both the sample and variant level QC are sufficient. And for more details about the QC of whole genome sequencing data, please go to poster number 193. Now, I'm going to walk you through some of the details of our sample level QC. So after sent to sequencing center, samples that passed the sequencing center QC were included in the March sample calling and returned back to us. We then performed another round of QC using the sample level metrics and created a set of high quality samples. And based on that, we conducted identity check such as ancestry and relativeness. And samples whose identity could not be solved were removed from the remaining analysis. Besides identity check, running PCA and IBD also allows us to generate principal components and genetic relationship matrix to control for sample structure in association tests. And out of this, we further pulled out a set of unique subjects by removing duplicates with relatively lower quality. And this constitutes the sample basis for many of the analysis later on. And out of these samples, 5041 also have the CFTR causal variant information from registry. And by using the whole genome sequencing data, we confirmed the causal variant for the vast majority of them. In addition, we were able to identify the missing causal variant for over 100 subjects and corrected the causal variant in registry for another 97 individuals. And for more detailed information about CFTR genotyping from WGS data, please check poster 202. Here is the results of the analysis that we did to sort out sample structure and identity. This plot provides you an overview of the genetic ancestry for all the CFGP participants relative to external reference population from one cell in the genome. So each point here is one sample, and the gray ones are those from one cell in the genome. What you can see here is the vast majority of the CFGP participants are in blue, which is self-reported white, and both the white and non-white CFGP participants clustered with their corresponding population from one cell in the genome. While the IBD analysis basically tells us whether the relationship inferred from genetic data matches to those from pedigree file. So the two x-axis here are the two metrics used to infer the pairwise relationship. So each dot here represents a pair of samples instead of a single individual. And only pairs that are at least slightly related to each other with kinship coefficient greater than 0.03 are shown up here. So in total, there are more than 13,000 pairs here, and most of these are from the twin and sibling studies because that's the only family-based cohort. And samples with unexpected relationship are either solved or reassigned with new identities, or if unsolvable, they would be dropped from the downstream analysis. So after the samples were cleaned, we conducted the variant level QC. Here is a summary. So in total, we received more than 120 million variants from the March sample BCF file, which is comprised of over 5,000 individuals. About 88% of those variants passed GATK VQSR, and out of those, another 88% were SNVs. After we applied hard filters on top of the VQSR, we were left with about 90 million SNVs that are considered as high quality. And here is the results of our concordance check for the SNVs that passed and failed the hard filters based on the 71 pairs of duplicates and MZ twins. The concordance was broken down by alternate allele frequency. And as you can see that the mean concordance is consistently higher for the variant that passed the hard filters, but was much lower for those failed. This suggests that most of the 2.4 million variants that are flagged by hard filters only were likely to be problematic. And here is a QQ plot from the GWAS of Marconi alias. We choose this phenotype for QC because it is highly heritable and readily available. And as you can see here, this is a typical QQ plot from a well-conducted GWAS. No early deviation from the reference line, with lambda being close to 1, suggesting that the population structure is well controlled, and also the variant filtration is likely to be sufficient. The huge deviation at the tail of this plot indicates there are many significant GWAS hits from this analysis. And for more details, you can go to poster number 192. So in summary, the CFGP is an extremely rich resource for studying CF traits and their genetic determinants at an unprecedented scale. So far, the project has created a number of interesting findings that you can check out at this conference. And finally, we would like to thank all the PI and the CF Foundation for the use of patient registry data. In addition, we would like to thank all the people with CF, care providers, and clinic coordinators for their contributions. And I will stop here and glad to take any questions.
All right, thank you, Dr. Ling. Wow, that's fascinating. So while we're waiting to look for questions to come through the filtering system, there's a lot of, I have a question for you. So, so what do you think what does identifying a modifier allow us to do? We know a lot about CF and CFTR, and we have CFTR modulators. Why are modifiers important? What can we do with that information?
I think there are two levels. So the first level is from the individual perspective. So with the identification of genetic modifier, at least currently we can stratify the risk for each individual with CF. And at the population level, I think it can help us to understand the pathophysiology of the CF traits, and also if there's any medication targeting those modifiers, those medication might work for all the CF patients, including those who may not have eligible modulators, which requires for certain CFTR genotypes.
Well, thank you. I can't tell if anybody's walking up to the mic. So I have another question for you. So there's, you sort of, okay, here's the first question. Dr. Ling, did the data take into account the therapies that the patients received?
We do have a modulator use information. So I think it depends on which phenotype you are analyzing. And a lot of the phenotype were taking multiple measurements. Some of the measurements were taken before the use of the modulators, and some of the measurements were taken after the modulator use. So there are many different types of analysis you can conduct based on what kind of questions you are interested in.
And you mentioned that this is as a resource. So what's the plan for this data being available to the CF community?
The current plan is we will make the data available in the future. The thought is to make it available in a mechanism similar to the CFF patient registry data through CF Foundation. So any PIs that are interested in applying for the data can propose for that.
Great. And another question, can you give us a sense of what kind of clinical data you know about the people in this study?
The clinical data, you mean the phenotype information or?
Yes.
So currently, we are investigating the genetic modifier for a number of phenotypes, like Meconium ileus, cystic fibrosis-related diabetics, response to modulator use, Pseudomonas infection, lung function, etc. If there's, we have access to the registry data, so we got a lot of phenotypes that can be investigated in the future.
And one more question, can you comment on, is the data powered to detect modifiers in CF patients of non-Caucasian background?
Currently, as PCA results have shown, there's 96% of our participants are self-reported as Caucasian. So I think the power will be relatively limited for non-Caucasian individuals. So this is one of the limitation, because 5,000 is a total number for this current study, given that CF is a rare disease. So the other way is, so another way to think about this question is, if the genetic variant affect both Caucasian and non-Caucasian samples, we will still be able to detect it at a certain level, because in the analysis, we'll be able to control the population structure. If there's any variant that only affect the non-Caucasian population, then we will have very limited power to detect that, I think.
Thank you. And I suppose unless it's a rare and high effect allele. There's another question, and then I think we'll move on after that question. Can you comment on how the identity of the individual sequenced was protected during this process?
Oh, this is a great question. So the confidentiality is always the most important issues. So we have gone long to maintain the confidentiality for both the clinical and genetic data. So all the samples were de-identified and were overseen by the CF Foundation.
Okay, great. And if anybody feels that they didn't answer that question, submit again, and we can circle back. And I think all these speakers are available over email or through the meeting chat facilities. Okay, thank you, Dr. Ling. Let's see, our next speaker is Marlu Bierlach, who is a PhD student at the University of Utrecht in the Netherlands, and we're really looking forward to hear about the organoid HIT-CF study.
Sorry, I think that went wrong. I'll start over. Thank you for the introduction. My name is Marlu Bierlach. I'm a PhD student in the Pediatric Pulmonology Department of the University Medical Center of Utrecht. And on behalf of all the co-authors depicted here below, I'm happy and excited to show you the first results of the HIT-CF organoid study. Not much to disclose, other than that this project has received funding from the European Union's Horizon 2020 Research and Innovation Program, depicted under this grant agreement. And in the next 10 minutes, I will tell you a bit about the background of the HIT-CF Europe project, tell you about our approach, show you, of course, some results, and tell you about our future plans. So this whole project is based on the problem that patients that carry ultra-rare CFTR mutations do not have access to approved modulator therapy. And if you look at this chart on the right side, we see that most patients carry a F508del mutation, either homozygous or heterozygous, and these patients have access to lumacaftor, ivacaftor, tezacaftor, ivacaftor, and also in Europe since last June, the triple therapy was approved by the EMA, which is very exciting. And then for the patients that carry gating mutations, we have IVA-CAFTR. But this leaves us with this group of patients on the top, the patients that carry ultra-rare mutations. And the goal for HIT-CF project is to get drugs available for these CF patients with these ultra-rare genotypes. And our approach is to use the intestinal organoid model as a predictive tool for clinical drug response. Here you see a simplified outline of the project, where we aim to collect rectal biopsies from adult patients in Europe and Israel with ultra-rare mutations, to culture these rectal biopsies into an intestinal organoid, which is a 3D tissue model where we can do drug testing on. And we will do these drug testings in screens to select the highest responders to those drugs and to invite them into clinical trials, which will be held within the ECFN's CTN clinical trial network and will be designed as crossover studies. So I think most of you are familiar with the organoid model, have heard about it, but for those of you who've never heard about it, I will explain the basics of the forskolin-induced swelling assay that we use. So here on the left side, you see organoids derived from a healthy control. And what you see, if you add forskolin to these organoids, after 60 minutes, they start to swell. And forskolin activates CFTR. CFTR is on the apical membrane in these organoids, and when activated, it secretes fluid into the lumen and the organoids start to swell. Here we see organoids derived from a patient carrying a homozygous F508del mutation, and what we see here, if we add forskolin and look after an hour, you don't see much change because of the mutation. But if we incubate these organoids with drugs, in this case lumacaftor, ivacaftor, and then stimulate them with forskolin, we do see swelling almost as big as in the control cells here. And if we then specifically inhibit CFTR and add the drugs, we don't see the swelling. And this shows us that the forskolin-induced swelling assay is a CFTR-dependent assay and can show CFTR activity. So HIT-CF has a big consortium with collaborating partners. There are 2 CF organizations, the ECFS CTN, CF Europe, 4 different laboratories, 3 academic ones in Utrecht, Lisbon, and Leuven, and 1 reference lab called The Hub. We have 2 pharma partners, Elox Pharmaceuticals and Proteostasis Therapeutics, and 2 supportive companies. And the first part of the project is the organoid study. We had inclusions up until last February, and we were able to collect 502 rectal biopsies from adult patients throughout Europe and Israel, all carrying ultra-rare mutations. And here you see all the countries that participated and the number of patients they included. And on the right 2 rows or columns, we can see the number of patients that will be screened into the 2 drug screens. We have 1 drug screen with drugs from Elox, and you see that the numbers they differ between the 2 screens, and that's because Elox has a read-through compound, and that's why we said these patients should carry at least 1 nonsense mutation, and for the PTI screen, it's the other way around. We say patients could carry a maximum of 1 nonsense mutation. And today I will show you the first results of the PTI screen. PTI proteostasis has 3 different modulators in their pipeline. The first is dirocaftor, which is a potentiator and enhances the ion transport. We have posenacaftor, which is a corrector that promotes maturation of the protein. And we have nezolicaftor, which is an amplifier. PTI already showed some phase 2 data on different congresses. And what we see here is that the amplifier has an additive effect on top of the corrector and the potentiator. And in the triple therapy, in a phase 2 study with 11 homozygous F508del patients, we see an increase of percent predicted FEV1 of 8% and a decrease of 29 millimolars per liter of sweat chloride. And then to our results, what we see here is a waterfall plot with the organoid responses of the first 228 patients ranked from highest to lowest responders to the combination dirocaftor and posenacaftor. And the organoid response here, you can see on the y-axis, is stated as the mean area under the curve corrected for DMSO response, and DMSO response is the response only to forskolin, not to drugs. And then they're ranked from highest responder to lowest responder. And here the dotted line is a reference line, and the reference line is the mean organoid swelling from organoids derived from a homozygous F508del patients and how they swell to the treatment of tezacaftor, ivacaftor. And what you can see here is that over 50% of these organoids from people with ultra-rare mutations show to some extent a positive response to the combination of dirocaftor and posenacaftor. And even this upper part here, that's up to 25%, shows a higher response than our reference line. And if we zoom into these high responders and look at how their response looks compared to the control, we saw some interesting results. So here I show you 31 samples that either show a high response, so an AUC higher than 1,000 to dirocaftor, posenacaftor, or a high response to tezacaftor, ivacaftor. So all the samples were measured to both combinations. And if we look here at the bottom right, we see that 15 samples only show a high response to dirocaftor, posenacaftor, and don't show a high response to tezacaftor, ivacaftor. And here on the upper left part, we see 6 samples that only show a high response to tezacaftor, ivacaftor, and here in the middle there is some overlap, and it shows us that there is a heterogeneity in response to these different modulators. And it leads us to the conclusion that a substantial proportion of patients that carry ultra-rare CFTR mutations show an in vitro response to the combination of dirocaftor and posenacaftor. Of these patients, once the screen is completely finished, we will invite the highest responders to participate in a clinical crossover trial to also show clinical effect and to further validate the organoid model as a predictive tool. And we saw that there is a heterogeneity in response to different modulators, which favors a personalized medicine approach. And with this project, we hope to lay the basis of this personalized medicine approach, which is especially useful for patients who don't have access to modulator therapy at this moment, or who will need to switch modulators in the future, either due to lack of efficacy or due to side effects. And we also are working on a European-wide biobank where we can store all these organoids for future use, either for drug development programs or patient selection for future clinical trials. I would like to thank you for your attention. I would like to thank my promoter, Kors van de End, and co-promoter, Jeffrey Piekman, and of course, all the colleagues that have worked on this project, which are a lot of people. Thank you, and I'm happy to take questions.
Thank you so much, Dr. Bierloch. While we're waiting on people to go up to the microphone, I'm wondering I noticed that you had an asterisk at about 1,000 for the effect of TEZ-IVA in your assay on Delta F508 homozygote organoids. Now TEZ-IVA has a little more modest clinical effect than TEZ-IVA Alexa. I'm wondering if you can relate magnitude of effect in the organoid assay to some kind of expected clinical effect on lung function or sweat chloride.
That's a good question. And I think it's first of all, it's very interesting to repeat these experiments in the future with, of course, in relation to the triple therapy. But I think that we have shown some, published some data on the correlation between clinical effect and organoid effect. And that correlates quite well. So I think what we can expect for these drugs, especially in the high responder group, is similar to a bigger effect than the tezacaftor, ivacaftor shows in patients with a homozygous F508del mutation.
Okay. We have a question from Dr. Steve Rowe. Interesting talk. Can you draw any conclusions about the mutations that were observed to have a response? And question 2, were there any common themes on CFTR genotype, or does this appear to be explained by things outside of CFTR genotype?
I think this is a question I can answer hopefully next year, because we are still blinded for the genotypes at this moment. Because we're gonna, for the first clinical trial, we're gonna invite patients with high response and patients that are random selected. And therefore we decided to stay as blinded as possible, and also during all the laboratory experiments, we decided to not know upfront which genotypes we're looking at. So for now, for me, it's still unknown which mutations are the high responders and which mutations don't respond at all. And sorry, the second question, were there common themes on the CF? Yeah, so that's, I don't know yet. I don't know that.
Very interesting. You might have said something about this. Were people recruited for a biopsy regardless of CFTR genotype, or was it only people with ultra rare mutations?
Only people with ultra rare mutations. Yeah. So there's no one single F508del mutation in this group of patients.
Okay.
And I think we got quite a big, based on registry data, there are around 1,800 patients that carry an ultra rare mutation like this, and we were able to collect biopsies of 502 of them. So we got quite a broad range of those patients.
And one last question: when you say ultra rare mutation, there are certainly mutations such as stop codons or splicing mutations that are not expected to be responsive. Are people with those mutations also included in this study?
Yeah, and so that's why we have two different pharma companies within our consortium. So we hope that the patients with the stop mutations can benefit from the next screen that will be performed with ELOX drugs that has a read-through compound. And for the splice mutations, they are put in both screens to see whether they could benefit from either of the two screens, so either of the two drugs tested.
Okay. And if there are no more questions, thank you very much, Dr. Bierloff. That was a wonderful talk. Very interesting data. I look forward to the follow-ups.
Thank you.
Okay. I will be our next speaker, and I'll, for the record, state that I was not involved in selection of the abstracts for this session. So thank you again to the meeting organizers. Let's see. There we are. My name is Scott Blackman. I'm in pediatric endocrinology and genetics at Johns Hopkins, and it is my pleasure to present this work on behalf of the CF Genome Project, which was described by Dr. Ling in the first talk as a multi-center study funded by the CF Foundation and run by investigators at the 5 universities shown on your screen. Again, I thank the meeting organizers for giving me the opportunity to speak to you today. I have no relevant relationships to disclose. And as you know, CF-related diabetes is one of the most important complications of CF outside of the lung, which occurs with an increasing rate as patients get older. The risk of CFRD is, of course, determined by CFTR. In those people who have residual function CFTR mutations and exocrine pancreatic sufficiency, the risk of CFRD is reduced by about a factor of 20. However, in the majority of people with CF who have severe mutations and exocrine pancreatic insufficiency, the risk of CFRD is still actually highly variable, with new diagnoses anywhere between age 10 and age 60. This variation in risk is due mostly to genetic modifiers, which are genes other than CFTR. Our prior work has demonstrated heritability of CFRD in terms of modifier genes and genetic overlap with susceptibility for type 2 diabetes. We also constructed polygenic risk scores that can be used to gauge the risk for CF-related diabetes. One of the genetic modifiers identified in our prior work is the SLC26A9 locus, which was the subject of a symposium this afternoon and was a previously known player in the CF field, and it's also a modifier of meconium ileus. So I'm showing two figures from our second CFRD genome-wide association study, or GWAS. 3 loci were identified with genome-wide significance and were replicated in an independent sample. SLC26A9 encodes a chloride channel that interacts with CFTR. TCF7L2 variants there also affect the risk for type 2 diabetes as well as CFRD, and may be acting by modulating expression of either TCF7L2 or other genes. And the third locus, PTMA, encodes prothymosin alpha, which is involved in oxidative stress, inflammation, and other pathways, but we have work to do before we figure out exactly how that's working in CFRD. The polygenic risk score adds up the genetic risk for CF across those 3 loci plus 15 other identified type 2 diabetes susceptibility genes. I'm showing the risk to CFRD to people with severe CF with exocrine pancreatic insufficiency, that is. The risk of CFRD varies as a function of this risk score. For example, at age 25, only about 15% of those in the lowest risk group have diabetes, compared to about 50% of the highest risk group. Okay. Dr. Ling described the CF Genome Project to you. It is further described in 7 other abstracts at this meeting. The work I'm presenting used 8 million variants with at least 1% minor allele frequency. Phenotype information was derived from chart extracted data and also from the CFF patient registry. And I'll call your attention to the Venn diagram. There is some overlap between the CF GP samples and our prior GWAS studies. The GWAS on the last slide used the 5,740 individuals. And in the next 3 slides, we'll test our prior GWAS findings for replication in those 1,681 who were not in prior studies. The 3 genome-wide significant loci have already been replicated prior to this work. Despite the small number of newly typed samples in this study, we found reasonable evidence of replication at 2 loci and weaker evidence of replication at the 3rd, although again, they had been previously replicated outside of this work. 15 other type 2 diabetes risk variants were identified by the previous candidate study. These were the ones that went into the risk score. And we found some evidence of replication for 12 of these with just the risk allele being in the same direction, stronger evidence for 6 of them with a p-value of less than 0.05, and evidence sufficient to confirm independent replication for 1 locus, which is after correction for von Ferroni correction. And that's CEBPB. And finally, we replicated the 18 SNP polygenic risk score in this second now independent population. And you can see that in this graph by age 30, about 80% have CFRD in the highest risk group compared to about 25% in the lowest risk group. The remainder of the poster discusses pleiotropy, which means gene variants that affect more than 1 trait. So I already mentioned that one CFRD modifier is SLC26A9, being also a modifier of the risk of meconium ileus. The most recent meconium ileus genome-wide association study, shown here, identified 5 other either suggestive or genome-wide significant loci, and they are labeled here. Each of these is interesting, as I'll describe on the next 2 slides. Sorry about that. So here, we ask the question, are any of these meconium ileus modifier variants associated with CFRD, given that we already knew that 1 was? So we already knew about SLC26A9, which is on the top row. That's associated with both CFRD and meconium ileus. And in fact, 2 of the other 5 variants had study-wide significant association with CFRD. That's PRSS1 and CEBPB, and each of these is interesting. So SLC26A9, a brief summary, is a chloride channel which interacts with CFTR, and we recently published a study demonstrating that SLC26A9 and CFTR tend to be co-expressed in pancreatic ductal cells, and that the risk variants for MI and CFRD are associated with lower expression of SLC26A9. And our colleagues also showed that these were associated with reduced immunoreactive trypsinogen levels in newborns. So this suggests that increasing exocrine pancreatic function may be protective against CFRD. There is a slightly more complicated story for the PRSS1 locus. PRSS1 encodes the cationic trypsinogen, which is processed into trypsin and is probably the most important digestive enzyme in the pancreas. The variants at PRSS1 are known to be associated with lower gene expression and a lower risk of chronic pancreatitis. So it seems that decreased expression of PRSS1 reduces the risk of CFRD. That actually suggests there may be a place for the age-old hypothesis that pancreatic autodigestion could play a role in CFRD to some extent. On the other hand, reduced expression of PRSS1, that is, the variants that are associated with reduced expression of PRSS1 are associated with increased risk of meconium ileus. So it works in the opposite direction. And as our colleagues in the International CF Gene Modifier Consortium speculated, this could be related to thickness of the meconium. So there's less known about CEBPB, which is a transcription factor, but one important thing I'll point out is that this is actually triply pleiotropic with the same variants increasing the risk not only of CFRD and meconium ileus, but also type 2 diabetes. It was recently noted that CEBPB can regulate the expression of claudin-4, which is a tight junction protein expressed in the pancreas. Now this is interesting because a related gene, claudin-2, is a risk gene for chronic pancreatitis. So we don't know the answer yet, but one of our early hypotheses could be that CEBPB could affect the expression of claudin-2. So in summary, the initial CF Genome Project analysis for CFRD has identified 3 pleiotropic loci which affect both the risk of CFRD and meconium ileus with a slightly different story for each. The directionality for SLC26A9 suggests that worsening exocrine pancreatic disease might increase the risk for CFRD and meconium ileus. The PRSS1 variants act in opposite directions for the 2 traits and lend some support to the hypothesis that relates protease activity to diabetes risk. The third locus is also a type 2 diabetes risk locus, but we have work yet to do to identify its mechanism of action. And I'd like to again highlight the CFRD polygenic risk score, which effectively stratified CFRD risk again in this study. I'd like to thank the CF Foundation most of all for having the vision to fund the CF Genome Project Consortium, which has had quite a few interesting findings in so far only 11 months since getting sequence data. I'd like to thank all the co-investigators in the CFGP for helping create a fantastic initial set of studies demonstrating what you can learn from whole genome sequencing in a large number of individuals with CF. I'd like to thank the CF Patient Registry for helping fill out our phenotype data. I encourage you all to visit the 8 other CFGP posters at this meeting. Thank you all for your attention.
That was a great talk, Scott. Thank you. So while we wait for those questions to come in, I'm curious. So I noted that you had several modifier genes, specifically ATP12A and SLC26A9, that are also highly expressed in the lung. Have those been identified previously as modifiers for lung disease as well? And if you said that, sorry, I missed it.
Yeah, Dr. Burkett, that's a great question. So we looked in our lung data and we did not find SLC26A9 variants associated with diabetes to be at least strongly associated with lung function. I will say that there is an occasional signal that is hard to decide if it's real, but weak if true. ATP12A, I'm pretty sure that we looked at that as well and did not see a signal for lung disease. But you know, one of our priorities is to look at, as we're doing in this particular set of work by comparing CFRD and MI, is to take the modifiers of one CF trait and then look specifically to see if there's evidence that they might affect multiple.
And then our first question coming in is, have you looked at type 1 diabetes risk loci?
Yeah, so Dr. Oxett was the first author on the paper that we put out in the spring, AKSIT, and we generated type 1 diabetes polygenic risk scores, as well as risk scores for a number of sort of important diabetes and other metabolic traits in the general population. So in other words, what this does is it asks the question, does genetic risk for type 1 diabetes or type 2 diabetes or the like, does that affect a person with CF's risk of getting CFRD? So interestingly, there was probably, I would just best describe it as a weak signal for type 1 diabetes, which we were not expecting. Definitely not anywhere in the category of type 2 diabetes. So I think there's a little bit of further work to go on that. There's ways that we have to improve the type 1 diabetes risk scores, especially now that we have whole genome sequence data.
Thank you. And then the other thing I was wondering about is, I think it's interesting that your data indicates some interaction between the pancreas and the intestine in terms of how the pancreas is affecting meconium ileus. Was your data specific to incidence of meconium ileus specifically, or did you look at later instance of like distal intestinal obstruction syndrome that appears when patients are older at all?
Yeah, that's a great question. So we defined meconium ileus as strictly as we felt that we could. So we went back to original chart data. Most of the patients with meconium ileus had surgery. There were a few that did not have surgery, and we scrutinized all the available data to convince ourselves that this really was a true case of meconium ileus. And as you might imagine, somebody who comes into CF clinic and says that they had meconium plug syndrome and they can't remember what it was or the like, we did have a pretty high bar before we declared somebody to be meconium ileus positive in this particular study. So your question was really about distal intestinal obstruction syndrome, and we have access to those data, both from our clinical chart extraction and from the CF patient registry. We did a heritability study on DIOS about, I want to say about 10 years ago now, and at that time we did not find evidence for heritability of the DIOS trait in the twin and sibling study, suggesting that if modifiers have an effect on that, it's either diluted out by, let's say, messier trait definition in adults than a fairly clean trait definition of meconium ileus, or that it could be true that that modifiers don't really have a huge effect on meconium ileus. One interesting thing that you might imagine doing is to look for meconium ileus modifiers specifically as modifiers of the DIOS trait, because there your bar for your sensitivity for detection is a lot higher since you're looking at very few variants. So that's another project underway.
That's great. Yeah, I'd be interested to know how that turns out. So I think that's all the questions we have. Any closing remarks for our session?
Yeah, I'd like to thank all the speakers, I guess all the speakers except for myself, for coming in here and sharing your work, which looks fabulous. I'd like to thank the audience for your attention, and I'd like to thank the meeting organizers for really pulling off what's so far been a great virtual meeting. Thanks very much, and Dr. Burkett for being such a wonderful co-moderator.
Great, thank you again to all the speakers. This was a great session.